Most people are smiling. So I think we have another exam coming up a week from today. People have already started asking me about review sessions, and I will do another review session. It will probably be on the weekend, but I haven't decided on that. I don't tend to do them the night before the exam because it's too much of a rush to get the video posted for everybody. So I will announce that probably next time, but maybe on Friday. Last time I got talking about the proteins involved in DNA replication, and I started telling you about different DNA polymerases. So I told you, first of all, that the major polymerase for replicating most of the DNA in a bacterial cell is known as DNA polymerase III. And it's kind of a complicated enzyme. It has a whole bunch of different proteins associated with it, and we're not going to futz with that. You'll sometimes see a DNA polymerase III called DNA polymerase III holoenzyme, H-O-L-O-E-N-Z-Y-M-E, all one word. So if you see that, don't be confused. I'm not going to call it that, but that's what some people will call that enzyme. I also pointed out that there was another DNA polymerase that was important called DNA polymerase II. And the function of DNA polymerase II, as I described it, I'm sorry, DNA polymerase I, II is a different enzyme. My mind's not working. DNA polymerase I has the function of removing those primers. So it actually chews out the RNA and then replaces that RNA with DNA. And DNA ligase comes in and sort of binds everything together to make the intact strand on the lagging strand. I want to spend a little bit of time today talking about another activity of most DNA polymerases. So I've already hopefully planted in your head that DNA polymerase is a pretty remarkable enzyme, whether we're talking about the polymerase III or the polymerase I. And there's even more amazing things to see about it. So this involves an activity that's associated with most cellular DNA polymerases. And you'll notice I keep saying cellular DNA polymerases. And you might say, well, what else is there besides a cellular DNA polymerase? And the answer to that is that there are viral DNA polymerases. So viruses oftentimes will have their own DNA polymerase. And that will actually become important as I talk a little bit about this activity that you see on the screen. So DNA polymerases not only have the ability to make phosphodiester bonds and to work very, very rapidly, but they also have the ability to check their work. Okay? They have the ability to check their work. And so one of the ways in which they check their work, or one of the ways in which a cell checks its work, is by having another enzymatic activity associated with the same DNA polymerase. So both DNA polymerase I and DNA polymerase III have this next activity I'm going to describe to you. Before I describe it, at least in terms of giving it a name, I want to describe to you or show to you what it does. So we can see here we've got a duplex of DNA. Okay? So we see a strand that's going 5' to 3'. So we know this is the strand that's being made as we go off that direction. And it's copying the other strand, which is in the 3' to 5', because that's anti-parallel, which is how the strands are oriented with each other. If I ignore for the moment there's an error there, that error there is there on purpose, but I ignore the error, I want you to think about this DNA polymerase moving along. This DNA polymerase moving along, it's moving along here. It sees a G, it puts in a C. It sees an A, it puts in a T. Sees a T, puts in an A. Sees a C, puts in a G. Sees a G, and puts in, let's say, another G. It's made a mistake. Okay? So this polymerase is moving along, and it's moving in the 5' to 3' direction, and it has just made a mistake. Okay? Well, there's two things that can happen. One is it can just continue along, ignoring its mistake, as you may have done when you learn how to type. You just went on and came back and saw how many errors you made over the period of time. Or it may stop and try to fix that mistake. So this activity that's associated with what I've just described, stopping and trying to fix its mistake, is something called proofreading. Something like you would do if you were an editor. And you would recognize a mistake, you would stop, you would fix the error so that it didn't stay in there. Well, how does that proofreading activity work? Well, the proofreading activity works as a result of an enzymatic activity known as an exonuclease. Now, you've already learned of one exonuclease, but I probably didn't tell you specifically what it was. And that was the DNA polymerase I. When it removed the primers, it had an exonuclease activity associated with it. It started at the 5' end of the primer, and it chewed inwards. And it's for that reason we call that activity, that is the removal of primers activity, a 5' to 3' exonuclease activity. Now, we don't see that activity associated with DNA polymerase III. We only see it with DNA polymerase I, which is why the polymerase I was removing the primers. I'm telling you now that both polymerase III and polymerase I, both of these, have the proofreading exonuclease. And the proofreading exonuclease starts at the 3' end of the strand being made, and it starts working backwards. So that means that this proofreading activity is a 3' to 5' exonuclease, because it's starting at the 3' end and working backwards in the 5'. Well, if the last base that got put in was a mistake, like this one is on the screen, then it's very easy to back up, remove that, and then once you've gotten it removed, let's say you chew back a few bases, then you just go forwards again. Okay? This proofreading activity associated with DNA polymerases improves the fidelity, that is the accuracy, of the replication by about 10 to 100-fold. Okay? Meaning it's catching, an awful lot of errors that the enzymes that don't have that activity, make. Now, even with that, a DNA polymerase is not perfect. We know that mutations happen. And there's a variety of reasons mutations happen, and I'll talk a little bit about that later today when I talk about repair. Okay? But suffice it to say that this activity is important for cellular polymerases. Okay? Cells have in their vested interest maintaining the integrity of the DNA as much as they can. Because mutations can lead to death, mutations can lead to uncontrolled growth, known as cancer. Okay? And so it's important that cells maintain the integrity of their DNA. Viruses, on the other hand, don't have that limitation. They don't have that limitation. A virus wants to survive over a long period of time, not for just one cellular life cycle. And a virus's long period of time consideration means that when it gets into a cell, it wants to make as many copies of itself as it can. And if some of those are slightly different in DNA sequence, then what that means is that the virus may have some of those that will be able to survive conditions that other ones may not be able to. What we see with a virus is evolution in process. Okay? Evolution happens because of mutation. And that virus is very well set up for it. Now, not all viruses have that property, I should point out. But I'm going to tell you some that have that property that make a lot of sense, that you've heard about. Okay? Everybody has heard of HIV. And HIV has its own DNA polymerase. It's an unusual DNA polymerase because it's able to copy RNA. Other DNA polymerases can't copy, that is, use RNA as a template and make DNA from that. But HIV has a DNA polymerase called reverse transcriptase. Reverse, just like the word reverse, transcriptase, Transcriptase. Reverse transcriptase is a DNA polymerase, but it can copy either DNA or it can copy RNA. Now that turns out to be important in the life cycle of HIV, which we're not going to talk about here. I'll talk about that later. But most importantly, the reverse transcriptase of HIV does not have proofreading activity. It does not have proofreading activity. It does not contain the 3' to 5' exonuclease associated with cellular DNA polymerases. That means that HIV makes a lot more mistakes than cellular DNA polymerases do. And all of you have heard of anti-HIV drugs that work for a while and then don't work. Why does that happen? It happens because the virus is mutating, and you give enough mutation and selection over time, and what will happen is you will evolve something that will work its way around the drug, and that's what HIV does. So you can see pretty clearly that mutation, in this case, is in the virus's interest to actually persist over time. So viruses not uncommonly have DNA polymerases associated with them that are much more prone to error. That make sense? Yeah. Because of what?
Our selection or case selection between viruses themselves, like the number of offspring and longevity.
I'm not sure I understand what you're saying.
Are mutations less in cells than viruses?
Yes.
Longer than viruses?
Okay, so it's a complicated answer to your question. Okay, so his question was: are the mutations associated with viruses more favored because they've got a longer cycle than cells? Is that what I'm? That cells have a longer cycle. Cells have a longer reproductive cycle, yes, but viruses are surviving longer than the organism itself. So if we think about it, it's kind of a double-edged sword. So a virus doesn't know how long it's going to be before it gets to infect another cell. It could be floating out in the environment for a long time, actually. So I won't say the answer is what you said, no. Okay? But viruses have a variety of strategies that they use. Okay? This is one. There are many other strategies that viruses have. But the beauty of viruses is we can see evolution happening. People say, oh, evolution doesn't happen. Well, you can see it happening with a virus all the time. You can also see it by virtue of the fact you have to get a flu shot every year. Viruses are evolving their way around your immune system. If you don't stay up on your flu shots, you're going to get the flu because a virus will figure a way out around it. So mutation works in the virus's interest. Okay. That finishes pretty much what I want to say about DNA replication. Okay? I want to turn our attention now to another phenomenon known as DNA repair. Okay? So DNA repair is also very important. Cells have these incredible systems set up to maintain the integrity, that is the sequence, the integrity of their DNA for the reasons I've just stated. So it wants to be able to copy a DNA as accurately as it possibly can. And the cellular polymerases do a very good job of that. But there are other things that can happen to DNA that will affect the DNA's ability to function properly, to be replicated properly, etc. I'll give you a couple of examples. So one example is something that some of you probably do all too much of. And I always chew on my class about this. Okay? And that is that you expose yourself to either chemicals or to radiation that will favor mutation. And they favor mutation because of alterations that they make to your DNA. Let's imagine you take a chemical and you bind it to your DNA to one of the bases. That's not an uncommon thing that happens. You bind a chemical to your DNA, and let's say it's this base right here. The DNA polymerase is used to going through and reading bases, and all of a sudden it's got this base with this thing sticking off of it. What does it do? Well, there's a variety of things it can do. And one of the most common things it can do is if it's not fixed, it gets confused and puts the wrong base in. You've just made a mutation. Okay? People are all too comfortable doing that. We know about smoking, and smoking causes lung cancer. And smoking causes lung cancer because cigarette smoke contains things called DNA adducts. And these are chemicals that are reactive with your DNA that will bind, just like I said, and screw up DNA polymerase. Okay? Now, What's happened in recent years, especially among people, more educated people, is that smoking frequency has gone way down. Okay? And that's very good. Some people still continue to smoke, and not for reasons that were necessarily under their own control. But smoking has gone down. But other things that we've done have gone way up. And they're just as stupid as smoking. Okay? They're just as stupid as smoking. Now, I'm not going to ask for a show of hands, but I'll wager you that there's a decent percentage of you in here who have been to tanning booths. Okay? Tanning booths are designed to tan you. And tanning has a certain benefit, and it has a certain detriment. And the detriment that it has is they use ultraviolet light, which is what causes tanning. And that ultraviolet light also causes formation of what are called thymine dimers. And thymine dimers act just like the adducts in cigarette smoke. They confuse the DNA polymerase. They screw up the polymerase. The polymerase says, I don't know what to put in here. I'm going to guess I'll put a G in here. And all of a sudden, you've got a mutation. So the same people who are worried about what might happen if they smoked are quite happy going to tanning booths. And I always get people that in this class, and they'll later confess and say, oh, I got all worried when you said that about the tanning booths. Okay, well, good. That's why I'm here. I'm here to chew on you and help you to improve your lifestyle. Well, let's talk a little bit about how the cell deals with these things. Fortunately, your cells have systems to try to repair this damage. Okay? The more you overload those systems, the more likely you're going to have a mutation that you can't recover from. Just like the DNA polymerase is not perfect, neither is the repair system. So the more damage you do to your DNA, whether it's through smoking or through tanning booths or other things like that, the more likely you are to have a mutation that's going to kill you. Okay, so this brings up the subject of DNA repair. So DNA repair is an important consideration. This shows a sort of a schematic way in which a cell could deal with a repair. Now, I'm going to show you some proteins that are involved in doing some of this stuff. But suffice it to say that DNA polymerase plays a role in this process because DNA polymerase has an exonuclease ability to remove bases, okay? And that can help if some of those bases are damaged, for example. Okay? So if a cell is able to make a nick, that is a cut between a couple of bases adjacent to an error, being able to remove some of those bases may be beneficial for the cell. Since it's a DNA polymerase, it also has the ability to fill in bases that it removes. So it can chew back, and then it can move forward with polymerase and fill in the gaps. And I hope you would remember from what I told you about polymerization yesterday and Okazaki fragments that DNA ligase would then join everything together at the very end of that. So that's a very simple scheme for repair. Yes? It can go back and forth. Uh-huh. So polymerase 1 or polymerase 3, because polymerase 3 also has an exonuclease, a 3' exonuclease ability. It can. But if the cut is made ahead of the problem, then it can move backwards, remove the problem, and then move forward. So it can. Well, that's what I'm getting ready to talk about a little bit. Okay. So I don't want to give you too much detail, but I will say a little bit that there are proteins that are involved in recognizing where the damage has occurred. Well, first let's talk about thymine dimers. Okay. So thymine dimers, I hope I've scared all the tanning booth people about, are adjacent thymines on the same strand. Not on separate strands, not a thymine over here and a thymine over here, but on the same strand. Okay, you've got two thymines in a row. And in the presence of ultraviolet light, what can happen is formation of this what's called cyclobutane or cyclobutyl ring. These are covalent bonds right here and here. Normally you see that there's no covalent bond between the adjacent cytosine. Between the adjacent thymines. Okay? This covalent bond causes the DNA to be less flexible. It causes the DNA to bulge. And bulges are problematic because DNA polymerases are really good at reading a regular structure of DNA. A bulge confuses the polymerase. Because of this, the thymine dimers are what we call mutagenic. They favor the formation of mutations. Okay? This shows a schematic, and Mariah asked about what proteins cause this to happen. Okay? Well, there are proteins, and I'm not going to hold you responsible for these names, but proteins called EXI nucleases that help to recognize, hey, here's a bulge. This is a thymine dimer. Let's get this piece of stuff out of here. So it makes a couple of nicks, one there and one there. Other proteins help to get rid of this problem. And then DNA polymerase, in this case, comes in and says, OK, I'm going to fill this in. DNA ligase says, OK, we're going to seal everybody together, and everything's done. Okay? Now, there are many different repair schemes. This is one repair scheme. Okay? When I showed you the proofreading, that's a repair scheme. It's recognizing an error. It's chewing backwards. It's replacing the stuff moving forwards. Yes? No. So the exonuclease recognizes and cuts. So in this case, the exonuclease is cutting right here, and it's cutting over here. So it's cutting on both sides of the damage. Yes, ma'am? Exonuclease is a part of the DNA polymerase. That's correct. So exonuclease, all right, the 3' to 5' is part of both the polymerase 1 and the polymerase 3. The 5' to 3', which removes the primers, is only a part of polymerase 1. Okay. All right, let's see. By the way, that I should also give that a name. Sorry, I should have given that a name too. So this removal here is called nucleotide excision repair. There's 3 different kinds of repair I'm going to talk about. Nucleotide excision repair. Okay. The second one I'm going to talk about is called base excision repair. Okay. And we can see in this case that there's been damage that's happened to a base. You might say, well, which one does the cell use? Does it use nucleotide excision? Does it use base excision? And there are different types of error that are dealt with by different systems. Again, that's way beyond what we need to talk about here. I want you to just understand what the systems are. So nucleotide excision repair, you saw it chopped out a section of the damaged DNA, and then a DNA polymerase and a DNA ligase fixed it, right? Well, this one is much more specific. We've got a damaged base, and that base itself actually gets removed. Damaged base, you can see there's the base. And look, something has come along. It's called a glycosylase. Again, I'm not going to hold you responsible for that. But it's removed the base without even doing anything to the backbone. Okay? Other proteins come in and say, OK, well, if we're going to repair this with a polymerase, we better break the backbone. So they break the backbone, leave a gap, and then DNA polymerase and DNA ligase comes in and does their thing. So base excision repair has at its very roots removal of a base, whereas nucleotide excision repair has removal of a section of a strand. Yes?
After the base is removed, what do you want to remove the backbone?
So this is called, well, it's a long name. Again, if I say it, I'm not throwing it out there so that you'll have to memorize it. So I'm just saying something comes along and chops that out. Yeah. Okay. My sort of rule, the reason I'm not saying some of these, is my rule is if I talk about it in class and people take that very literally, then if I say the name, people say, oh, I've got to know that name, right? So if I don't say the name in class, then those same people will say, okay, well, I don't have to worry about that. So I'm trying to keep it simple. But anyway, this is it's called an apyrimidase, which in this case cuts out a pyrimidine. There's also apurinases that will also cut out purines. Okay, that's DNA. That's base excision repair. The last one I'll talk about is related, actually, to the proofreading. It's called mismatch repair. Okay. Now, mismatch repair can happen as a result of a couple of things. Right? mismatch repairs suggest, hey, there's just a mismatch here, right? I've got a G where there should be a C. Well, if I have the wrong base in, what happens? I get a bulge that appears on both sides. Okay? And that bulge that appears on both sides tells the cellular machinery that, well, something happened here. Either there was some chemical change that converted, for example, a T into a C. That's possible. Okay? Or it's possible that the proofreading of the DNA polymerase missed something. And that happens occasionally as well. So it doesn't matter how it got there. What matters in this is that the cell recognizes that there's a mismatched set of bases. You don't see any damage here. There's no chemical damage. You just have the wrong base in the DNA. Well, with this system, we can see pretty readily what happens, what the cell does. Okay? It's got some proteins, mut L, mut S, mut H, which I shouldn't have said those. I'm not going to hold you responsible for them. Okay? Just take back what I just said. All right? That come in, they recognize there are 3 proteins. They recognize the mismatch. They chew away, just like we've seen before, and they allow DNA polymerase to come in and fill the gap, exactly like before. Okay? So even though they are 3 different repair systems, they all have common features. Recognizing, chewing away, and filling in. All 3 systems have that. Yes, sir?
Just chewing away the backbone, or is it also taking away the complementary mutation?
So it's just chewing away this one damaged strand. Because if it started taking these away, it wouldn't know how to repair. Right? Yes?
Why is it that in the first method, in this one, it chews away so much, rather than like the second one and the second one out? There's only one mismatch.
Okay. So these are all schematic drawings in the first place. So I don't want you to take them too literally. In the case of base mismatch, base excision repair, which is the second one, which is the one I think that you're referring to, generally you're talking about a very localized kind of a damage that's happened. In any case, why something would be this way is simply that's the way the system evolved. So we don't have good answers sometimes for why that's happened, but it has evolved this way that the repair system works that way. Yes?
How does it know which one was the wrong one?
Oh, that's a very good question. So Mariah wants to know how does the system know which one's wrong? Because if I randomly say this one's wrong, I'm wrong half the time, in which case I've let the mutation go, right? Which isn't a good career move. Okay? I'm going to tell you very briefly the answer to your question, but again, I'm not going to hold you responsible for it. Okay? I don't want to get Mariah in trouble here, otherwise everybody start hating her for what I'm going to make you responsible for. Okay, so you can sit back and listen to this. The cell has to be able to know which strand had the mismatch. And the cell makes one assumption. The cell assumes that the newest strand that was made is where the error was, because presumably the other strand got checked when it got done, right? So that's a reasonable assumption, that the newest strand that's made is where the error arose. So the cell then has to be able to figure out which strand is the newest strand. And bacterial cells have a very cool trick for doing that. They use a system called methylation. You see that CH3 right there? That's a methyl group that the cell puts onto the C within a GATC sequence that occurs in E. coli. Okay? That occurs anywhere. But E. coli recognizes that sequence and puts a methyl group on the C. It's kind of a way of like a gang, where you go and you put your stamp of your gang to indicate this is my neighborhood, right? Well, you put that on that C every time you have a GATC. It distinguishes that GATC, right? And GATC is a symmetric sequence. So, for example, this, I'm thinking of a eukaryotic system. In the prokaryotic system, it goes on the A. In the eukaryotic system, it goes on a C. But in any event, it goes on to, in this case, an A. Well, look, there's an A and a GATC in the other strand, too, right? So wherever there's a GATC, the cell's going to have a methyl A on one and a methyl A on the other, right? Except for newly synthesized strands. the A's haven't had a chance to get methylated. So a newly synthesized strand will not have a methyl on the A, whereas the old strand that existed before will. Because of that, the cell then understands which strand is the new one and says, oh, this must be where the error is. Let's chew this out. Now, the other answer to your question back over here, why does it chew out so much, is because GATCs, on average, only occur about once every 256 bases. So it's got to chew out enough, okay? It's got to recognize where the GATC is. It's got to chew back to the next GATC, right? And it's got to remove that error as well. Pretty good stretch. Yep. Yep. Mm-hmm. Not the backbone. Backbone is there. That's the backbone right there, right? It's chewing out the strand that's got the problem. Remember, we got two strands here, right? Clear as mud? Anybody awake? I'll assume everybody who's smiling is awake, and everybody who's not smiling either doesn't like my humor or is asleep. I'll assume it's the first, not the last. Okay. I'm going to show you something here. I talked about methylation, so this is a place where I will hold you responsible for something. And you see three different pyrimidine bases. You see thymine, you see uracil, and you see cytosine, right? Okay. If I methylate cytosine, you'll notice that this is what happens in eukaryotic cells. If I methylate cytosine, this guy looks like this except for that part, right? In other words, methylcytosine is just like this except for they've got a difference there, right? Why do I care about that? Eukaryotic cells methylate their cytosine as a way of controlling the expression of genes, except for the problem is that cytosine is chemically unstable. It's the least stable of the bases in DNA. What will it do? It will spontaneously, at a low frequency, go from here to here. Losing this and gaining this. Now, if that cytosine had a methyl group on it, what has just happened? We've gone from a C to a T. Right? A methylcytosine that spontaneously gains this oxygen becomes this, not this. A C to T mutation is definitely a mutation, right? And so we could imagine that the cell would have some, shall we say, concern about that. And this is one of the reasons cells have a mismatch repair system. Because you have in every cell in your body right now this thing going to this thing. And if that's not fixed, there's a problem. Now, the beauty is that you have now made, instead of a GC base pair, you've made a GT base pair. And your mismatch repair system recognizes that and fixes it. Let's imagine we didn't have a methyl group on that cytosine. Let's imagine that that cytosine just spontaneously gains that oxygen. We've now converted a C into a U. And guess what? U appears in DNA occasionally. Okay? What's U going to pair with? U is going to pair with A, not with G. So again, if we see this U-G base pair, again, we know that this error has happened, that this chemical instability has happened, and that the cell has a way to fix it. In both cases, the mismatch repair system is important. And in this case, it turns out that this can actually be fairly easily removed by the base excision repair, because it only has to chop this one thing out. Don't get too confused with that. Questions? Okay. So the methylation of DNA bases causes some complication. There you can see that happening, cytosine to uracil. Okay. I want to spend just a little bit of time talking about replication of DNA in eukaryotic cells. Because I alluded to this yesterday, there's a fundamental difference between prokaryotic and eukaryotic DNA replication, because in eukaryotes the DNA chromosomes are linear, and in prokaryotes they're circular. In prokaryotes we can completely copy the circular chromosome of a prokaryotic cell intact, no problems. We can't do that in eukaryotes. So let's understand what's happening here. Here is a linear eukaryotic chromosome. And here is an origin. That's what that OR is. An origin is where replication is going to start, and it's going to continue, right? So we see replication happening from that point. And don't worry about all the proteins and all the stuff on there. I'm just wanting you to envision for a moment that linear chromosome replicating. Okay? Let's think what's happening at the end of a eukaryotic chromosome. At the end of a eukaryotic chromosome, we've got a 3' and a 5', both at that end, right? And how does replication start? Replication starts with a primer that's an RNA molecule, right? That gets put on, and then polymerization happens, right? Let's imagine that I actually started replication, let's say, I don't have a good figure here. Hold on. Let me get a better figure for you. Okay, this is a better one. I've started replication going 5' to 3'. Here's my RNA primer. I'm at the end of the chromosome, and leading strand synthesis is going to go here just fine. Okay? How do I fill this gap in? Because if I have something like DNA polymerase I that comes along and starts chewing in, it's got to have something behind it to start from, because it needs a primer to start, right? It can only extend an existing piece of nucleic acid, but there's no piece out here. And the answer is it doesn't. It does not replicate that. It means that at the 5' end of each eukaryotic chromosome, every cycle of replication, a piece of it is lost. And when the replication comes back the other way, it can only go as far as right there. And so now I've got a double-stranded DNA that's missing that much. How do we live? Well, the way that we live is the end of our chromosomes in eukaryotic cells has a repeating sequence, a short sequence of about 10 nucleotides that's repeated thousands of times. Thousands of copies, one after the other, after the other, after the other, after the other, after the other. People used to refer to that as junk DNA. They didn't know what the heck it did. It didn't code for any protein. And so if it didn't code for any protein, as far as they were concerned, it didn't have any function, therefore it was junk. Well, the fact that it didn't code for protein was one of its strengths. Because let's imagine, if you will, that I had an important protein that was coded at the end of my linear chromosome. After one cycle, if the cell needed that protein, the cell just died. Not a good career move, right? On the other hand, if I have a repeated sequence that doesn't do anything except repeat, then every time the cell divides, I lose a little bit, but the cell doesn't care because there's no protein that was there. It loses a little bit more next time. It loses a little bit more next time. It loses a little bit more next time. Everything's fine and dandy, right? So the telomere protects the cellular coding regions. It protects the cellular coding regions. It provides, as it were, a buffer against loss. Yes? I'm sorry? Prokaryotes don't have telomeres because they're circular. That's correct.
Does the lost DNA ever get recycled?
Does the lost DNA ever get recycled? No, because it's just chewed up by the exonuclease, and it's mostly coming as RNA anyway. So it's not recycled. Yes?
When telomeres are about used up,
Yep.
start getting into the useful proteins, does that happen in humans in the 30s, 40s? When the
Okay.
cells start to-
So when, this isn't infinitely long, is his question. When we start getting into the goodies, as it were, what happens? The cell starts to die. Yep. So one of the things that happens is people say, well, is it possible that the length of my telomere determines how long I myself will live? And there is some evidence that that may be the case. Okay? There's some evidence that the length of your telomeres is linked to longevity. It's not a perfect relationship. There's complicating factors. Okay? But that may be the case. Yes?
Why do the ends get chopped off?
Why do the ends get chopped off? Because the way they got started in replication, they started out as an RNA primer at the 5' end.
Don't they just start in the middle?
Well, ultimately, this one is going to have to start from this end, because even if it started in the middle, that would only take care of replicating going that way. It wouldn't take care of the end. So I have to start at the end if I'm going to cover this part of it right here. In other words, I can't make things 3' to 5'. I've got to make things 5' to 3'. Okay? So starting something at the end has got to happen. Yes? I shouldn't have showed you that origin in the middle. Okay. There are origins in the middle. But remember I said eukaryotic cells have hundreds of origins because they're so big. So here's one at the very, very end of the molecule. Okay? Okay. Now let's think about this. If you think about this, you say, well, if the length is related to how long I will live, then that means my kids aren't going to live at all. Right? Because they're going to have even shorter telomeres than I do. Right? So how do my kids have a chance? Well, it turns out that telomeres are put on to DNA, okay, starting at the process of fertilization. There's an enzyme called telomerase, Telomerase. And telomerase's job is to make telomeres. And all it does is it copies the same 10-base sequence over and over and over and over and over. The longer telomerase works, the better off you are, probably. Okay? Now, telomerase is also a DNA polymerase. And telomerase is an unusual DNA polymerase. It carries the thing it copies. It carries the template. And the template is a little 10-base pair piece, or 10-base piece of RNA. It's carrying a 10-base piece of RNA that it copies over and over and over and over. That means that telomerase is also a reverse transcriptase, something that copies RNA and makes DNA. Now, when we look in cancer cells, we discover something interesting. Most cancer cells, okay, have decent-sized telomeres. And this sort of makes sense. Because if they didn't have a decent-sized telomere and they kept dividing and dividing and dividing, what would happen to them? They would die, right? Turns out that telomerase, okay, is active in cancer cells. Telomerase is active in cancer cells. This is the reason why we probably don't want to activate telomerase in the rest of our cells. Those cells, the cancer cells, are called immortal because they will keep going. If you grow them in a laboratory, you can keep them growing forever. You take a normal cell and you try to grow it in a laboratory, and it'll grow for a few generations, and it will die because it will run out of telomeres. Okay? So one of the approaches to dealing with to dealing with cancer is actually to design telomerase inhibitors. And there are some drugs that are in testing right now that look promising. It's not the cure for all cancer, but it looks like it may be helpful against certain types of cancer. You had a question?
It's kind of based on that, but also it's kind of random. But is that what Henrietta Lacks did?
Okay, so Henrietta Lacks was the woman for whom they took the cells called HeLa cells that they propagate in the laboratory. She simply had a uterine cancer cell or ovarian cancer, can't remember, that they took from her. So I'm not sure what the question was. Those cells are immortal, yes, yes. So virtually any cancer cell is immortal, yeah. So Henrietta Lacks, a very interesting story, the woman that they got a cell line that the vast majority of modern human cell research has been done on over the past 50 years. Her name was Henrietta Lacks, and somebody did a very interesting book on her just recently. Okay, I'm running out of time. Other quick questions? Yeah.
So, really quick, so the telomerase, that's kind of like the cause behind cancer.
Telomerase isn't a cause behind cancer, but it is commonly associated with cancerous cells, yes.
So, I was kind of confused. So, the telomeres, are those added like to cells at all times? Like,
No. So they're added during the time after you've been fertilized, okay? After your mom's egg got fertilized, telomerase became active. They're also found in stem cells, which is a little complicating, okay? So that you have stem cells that are regenerating things in your body and so forth. So it's not like you only have one set of telomeres that you can live with and then dies. That's why it's not a complete relationship for how long you'll live with your telomeres. Those in cancer cells, those three types of cells, the fertilized egg cells, the stem cells, and the cancer cells are where you find the telomerase. Okay, that's been a long one today. Let's have a quick song. This is another one I can't sing worth a crap, so I will have somebody else sing it for me. And here we go. Making DNA for me.
Replicating DNA, proofreading the enzymes QC path, choose that from the three. I can't have a G paired with T, so repair it, please. Chem damage is concerned too, 'cause it can cause mutations inside you. When dimers stem from sunlight UV, fix the DNA for me. Such pathways of excision cause cells to have to make a decision. Should they go straight ahead with repair, or take themselves right out of there? Then lastly, there's recombination. Swap strands readily. Crossover homology.
Okay, guys, see you tomorrow.